There once was a land home to countless flocks of sheep, and a shepherd that cared for them better than little Bo Peep. They spent their days eating and bleating and brushing their fleece. If you ask nicely, they may repeat the old shepherd's tale of the great beasts. Beware of the creatures that come in sheep's clothing. They may appear like the others and seem unimposing, but once they undress, the truth will reveal wolves with great gnashing teeth that prefer lamb over veal. The wolves' torment continued as their numbers increased. The shepherd's only option was to poison the beasts. When the wolves fell, so did some sheep. It turns out the flock was not immune to life's last eternal sleep. But some of the wolves didn't fall for the trick. They outsmarted the shepherd with their quick wit. They had had enough and decided to leave. There were still plenty more flocks in the land to deceive.
Whenever prostate cancer spreads from beyond the prostate, whenever it metastasizes, that means that you'll probably die from it. Prostate cancer primarily goes to the bone, and it actually goes from your bones to your viscera, to your soft tissues. When a man presents with metastatic disease, they undergo what's called androgen deprivation therapy, which is basically a nice way of saying chemical castration. Pretty much all men, after enough time, develop what's called castration-resistant prostate cancer. And this type of prostate cancer we're now seeing, it's completely testosterone independent. And this disease is very aggressive. There's no good way to actually treat this super aggressive variant of prostate cancer.
There must be a better way to determine friend from foe. The shepherd thought for a while, then exclaimed, I know. If I pick out the traits that separate sheep from beast, like long tails, sharp claws, and gnarling teeth, I can poison the devious creature that lies beneath. Imagine if wolves could be identified from every flock in the land. No longer would innocent sheep die in our hands. This tool would prove quite useful, I really must say. Now I just need to make it, somehow, some way.
There are very few effective immunotherapies for prostate cancer. In fact, there aren't any real immunotherapies for prostate cancer. By definition, if you make an antibody, that's an immunotherapy. And our belief is that these super aggressive prostate cancer cells express unique antigens on their surface. And our goal is to identify these antigens and target them with antibody fragments using antibody phage display.
Antibodies have the potential to be incredibly useful tools for targeting prostate cancer. The target antigen should be more strongly associated with a cancer cell than a healthy cell. For example, the shepherd should choose a trait that only the wolf possesses, such as its sharp teeth. Biopanning is an in vitro selection process to screen for antibody fragments with high affinity for a target antigen. In the biopanning process, the target antigen is immobilized to a solid surface and antibodies are selected from human antibody phage display libraries. These libraries contain antibody fragments that are displayed on the surface of M13 phage, with the corresponding antibody gene packaged in the phage particle on a separate plasmid. Some fragments will bind non-specifically to the solid surface or other phage, and are washed away so that only antibody fragments selective for the target antigen remain. These selective antibodies can also be tested in a cross-reactivity assay to remove any fragments that have affinity for an off-target antigen. This whole process is repeated several times to enrich the population for strong binders, from which functional antibodies are engineered.
When you make an antibody, you can do a lot of different things with it. You can use it for imaging purposes. You can attach small molecule drugs to it for what are called antibody-drug conjugates. They're all the rage right now in biotechnology. You can also attach therapeutic radioisotopes to antibodies for radioimmunotherapy. One thing that I'm currently getting into is taking antibody fragments and using them to engineer natural killer cells to make chimeric antigen receptor natural killer cells.
Previous work by Dr. Aaron Lebeau and colleagues led to the discovery of an active site binding inhibitory antibody termed U33 from a human antibody phage display library. The U33 antibody recognizes urokinase plasminogen activator, or UPA for short. UPA is a protease that is not normally expressed under healthy conditions. It's mainly secreted by very aggressive forms of prostate cancer, including the late-stage type that the Lebeau lab is interested in.
What's really cool about U33 is it leads to internalization of the U33 UPA complex. This U33 bound to UPA and got sucked into the cancer cell at a very high rate. So as a result, we had really beautiful scan quality in our in vivo experiments with mice. And because U33 can gain entry into cancer cells, that means that it's potentially great for therapy.
The shepherd brought his tool to the pastures, far and wide, to find where the creatures were trying to hide. It worked quite well, and was easy to tell where the wolves were trying to dwell. As the poison seeped into their veins, not a single beast remained. The sheep cried out in relief, "Hip, hip, hooray! We are saved, the wolves have been conquered today." Now if the wolves ever try to come back, the shepherd will have a plan of attack. For now he knows how to tell friend from foe.